Unlocking the secrets of cellular dynamics with cryo-optical microscopy. A revolutionary approach. Imagine being able to pause time and take a closer look at the intricate dance of cellular activities. This once unattainable dream is now a reality, thanks to a groundbreaking technique developed by researchers at the University of Osaka and collaborating institutions. Published in Light Science Applications, their cryo-optical microscopy method offers a high-resolution, quantitatively accurate snapshot of dynamic cellular events at a precisely chosen moment. The quest for accurate observation of high-speed cellular dynamics at high spatial resolution has long been a daunting challenge in optical microscopy. Striking a balance between temporal resolution and photon budget, the amount of light collected for the image has proven elusive. Limited photons result in dim, noisy images, obscuring crucial spatial and temporal details. In a bold move, the research team decided to freeze the entire scene rather than chase speed in imaging. They developed a specialized sample freezing chamber that combines the benefits of live cell and cryofixation microscopy. By rapidly freezing live cells under the microscope, they captured a frozen moment of cellular dynamics with remarkable clarity. Consider the case of calcium ion wave propagation in live heart muscle cells. The team froze these waves and observed their detailed patterns in 3 dimensions using super-resolution techniques, which are typically too slow for capturing fast cellular dynamics. This innovative approach offers new insights into life science and medical research, opening doors to understanding the mechanisms behind dynamic biological processes. Lead author Masahito Yamanaka highlights the technique's ability to preserve both spatial and temporal features of live cells through instantaneous freezing. This allows for precise quantitative measurements using various optical microscopy tools while the cells are immobilized. The team also demonstrated improved quantification accuracy by freezing cells labeled with a fluorescent calcium ion probe and using exposure times 1,000 times longer than in live cell imaging, significantly enhancing measurement accuracy. To capture transient biological events at precise moments, the researchers integrated an electrically triggered cryogen injection system. This system enabled freezing of calcium ion waves at a specific time point after the event's initiation, with 10 millisecond precision. This level of temporal accuracy allows researchers to observe transient biological processes in unprecedented detail. Moreover, the team explored the potential of combining different imaging techniques, which are often challenging to synchronize. By freezing samples nearly instantaneously, they could apply multiple imaging modalities sequentially without temporal mismatch concerns. In their study, they combined spontaneous Raman microscopy and super-resolution fluorescence microscopy on the same cryofix cells, revealing intricate cellular information from multiple perspectives at the exact same point in time. This revolutionary innovation paves the way for observing fast, transient cellular events, providing researchers with a powerful tool to unravel the mysteries of dynamic biological processes. The technique's versatility and accuracy promise to revolutionize our understanding of cellular dynamics and contribute to advancements in medical research. For more information, visit Time Deterministic Cryo-Optical Microscopy, Light Science Applications, 2025. DOI 10.1038/S4377/01941. https://dx.doi.org/10.1038/S419/01941. This document is subject to copyright. Reproduction in whole or in part without written permission is prohibited. The content is intended for informational purposes only.